Today, I am going to explain various advantages and limitation of different bioreactors and an overview about recycle reactor. The contents of this lecture include an introduction to bioreactor, bioreactor design, summary on chemostat bioreactor, problem associated with the chemostat bioreactor, advantages and limitation of various bioreactors, treatment bioreactors, definition for recycle reactor, requirements for the designing of the chemical reactor, advantages and disadvantages of recycle reactor, and finally the application of recycle reactor. First, I am going to give definition for bioreactor. Bioreactor refers to certain devices or system which support biologically active environment where chemical processes are undergoing which involving organism or their derived biochemically active substances. The bioreactors can have any shape according to the application. Also, the processes inside vary as for the environment to be limited differs. For example, aerobic and or anaerobic environment cycles. Bioreactors also use devices and or system meant to grow cells or tissue in the context of cell culture. In the last class, I have clearly explained what is the bioreactor and what are the types of various bioreactor. However, here I am giving a short summary on designing of bioreactor. Under optimum conditions, there can be use of microorganisms or cells which are able to perform normal and or with additional modified function. Even small changes in the environment affect functions as reaching the critical threshold that lead to major changes in the bioreactor. For example, gas which may be air, oxygen, nitrogen and carbon dioxide. Then flow rates, temperature, pH and dissolved oxygen level. Also, agitation speed need to be closely monitored and controlled. Thus, overall bioreactor design should consider details such as limitation on the production of hydrogen by proton gradients, carbon dioxide and requirements for carbon binding. A bioreactor therefore consists generally of a vessel, sensors for growth conditions and network system. Depending on how they are designed and operated, they can be classified as batch bioreactor, fed batch or continuous bioreactor. One such continuous bioreactor is also called as chemostat bioreactor. In this type of bioreactor, where the fresh medium containing growth factor is fed into the cells at a constant rate. In order to maintain the constant bioreactor volume, the waste media is made to leave the reactor at the same rate to control the rate at which the cells grow. It is referred to as growth limiting factor. Then we could discuss some problems associated with the chemostat bioreactor. Some of the problems associated with the chemostat bioreactor include foaming. It is nothing but when feeding the fresh medium, some sort of foam can be appear on the top of the liquid. This can deprive the cells of oxygen and affect the microbial growth. Then the cells present in the bioreactor can be ruptured during the process of aeration. Also, some cells may adhere to other surfaces or grow on the walls of the bioreactor. Also, in some cases, the medium may drip into the chamber rather than flow. This dripping causes pulses and fluctuation in the nutrients, thus affecting the operation of the chemostat bioreactor. However, some of the problem above can be limited by the use of anti-foaming agent in order to reduce the effect of foam formation in the bioreactor. Then, aeration and conditioning of the cell should be gentle. Next, if dripping of the medium is inevitable, drops should be made very small so as not to cause fluctuation. Then, we can also use vessels with larger volume in order to reduce the effect of drip during the fermentation reactions. Next, we will see some advantages and limitation of different bioreactors. First, batch bioreactor. The advantages of batch reactor are it is suitable for small scale production. suitable for process where different range of product are able to obtain in the same equipment. Then it is suitable for long taking reaction and suitable for reaction with superior selectivity. Next, the limitations are the batch reactor is not suitable for long batch sizes, and commonly the batch reactor is closed system in which once the product is entered only it comes out as a product after the completion of the reaction. Next, continuous stirred tank reactor. The advantages include it is highly flexible device, reactants can be easily removed during the reaction, it is economically beneficial to operate in series and parallel. Then the reaction can be carried out in horizontal as well as the vertical reactor. The major advantage is continuous operation. We can operate the bioreactor in continuous state. This type of reactor having good temperature control, easily adapt to two-phase runs and good control. Also, it is simple to construction, low operating cost and easy to clean. These are the some advantages of continuous stirred tank reactor. Then we will see some limitation of continuous stirred tank reactor. Number one, it is more expensive and complex than tubular units. At steady state, flow rate must be equal to the flow rate out. Otherwise, tank will go empty or overflow. Then, bypassing and channeling possible with poor agitation. These are the some limitation of continuous stirred tank reactor. Then we will see plug flow reactor. The advantages are it is highly efficient than both at the same volume. Then the plug flow reactor have several pipes or tubes in parallel. Both of the operation for vertical and parallel are same. In this type of reactor, both of the operation for vertical and parallel are same. They can be easily jacketed and reagents can be introduced other than the fixed inlet. The limitation is it is not economical for small batch fermentation process. Next, the advantages of continuous stirred tank reactor when performed in series or parallel. When high conversions of reactant are needed, several continuous stirred tank reactor in series or parallel can be used. In these cases, equally good results can be obtained by dividing a single vessel into compartment while minimizing back mixing and short circuiting. Then the larger the number of continuous stirred tank stages, the closer the performance approaches that of a tubular plug flow reactor. The last thing, advantages of plug flow reactor when performed in series or parallel. Tubular plug flow generally is preferred to laminar airflow because mixing and heat transfer are improved for slow reactions and especially in small laboratory and pilot plant reactor. Establishing tubular flow can result in inconveniently long reactors or it may require unacceptably high feed rate. Next thing, treatment of bioreactors. Bioreactors may have a fluid suspended medium or a solid through which the fluid is circulated to support the cell growth. In the suspended growth system, organic matter is degraded by bacteria to produce carbon dioxide and biomass which is recycled back or disposed of as sludge. For bioreactors which have a solid support growth medium, the microorganisms are grown on a film of the supporting structure. This bacteria degrade the water or fluid as it passes through the solid matrix. This degraded water then helps in degrading the waste which are present in the solid matter. Next, the oxygen is very essential in aerobic process and keeping an oxygen supply of oxygen can be a difficult task because of its poor solubility in water. Oxygen supply can be improved by constant and uniform mixing or stirring which also helps to mix nutrients present in the process. This process however has to be done at certain rates or speed in order not to damage the organism or cause unnecessary alteration in the reaction system. For cells to survive in the bioreactor must be maintained at favorable temperature, gas composition and correct growth medium and factors such as pH, glucose concentration, and other nutrients which are needed for the growth of microorganism present in the bioreactor. Some cells need to be attached to a surface for them to grow, while others can grow in suspension. However, it is possible to tailor the cells to grow in suspension as to increase the density, which may not be possible in adhesive surfaces. So, if we want to get product in good quality and good quantity, we should maintain the optimum conditions for the microbial growth. Next, recycle reactors. Recycle reactors are used when the reaction is autocatalytic or when it is necessary to maintain nearly isothermal operation of the reactor. In certain situation, it is found to be advantageous to divide the product stream from a plug flow reactor and return a portion of it to the entrance of the reactor. In a recycle reactor, the product stream from a plug flow reactor is divided and a portion is returned to the entrance of the reactor. Let the reaction ratio R be defined as volume of fluid returned to the reactor entrance divided by the volume leaving the system. This type of recycle ratio can be made to vary from 0 to infinity. Then the reflection suggests that as the cycle ratio is raised, the behavior shifts from plug to mixed flow. Thus, the recycling provides a means for obtaining various degree of back mixing with a plug flow reactor. From a plant-wide perspective, the design of recycled chemical reactor should comply with the following requirements. Number 1. Ensure optimal use of raw materials in a proportion as close as possible to theoretical stoichiometry so that accumulation of reactant and degradation of selectivity is prevented. The second thing, offer stable and safe operation over a long production rate and product specification. Third one, allow stable and smooth transition between various operating points. It may be observed that above requests regard not only the reactor design but also make up strategy of reactant and in general the plant-wide control of the material balance. It is obvious that the large variation in flow rate and composition in the reactor outlet could upset the operation of downstream unit. Next, advantages of recycle reactor. The recycle bioreactor has two major advantages. Number 1, less time required for desired conversion. That means when compared to other bioreactor, the recycle reactor has short time for the conversion of substrate to product because the recycle reactor has special components than compared to other bioreactor. That is back mixing. This back mixing helps to convert the substrate into product in a short time. Next, we can get different percentage of conversion by only changing the recycle ratio. This point clearly stating that by changing the recycle ratio, we can obtain the different percentage of product. We know to change any feed supplement, pH, temperature, oxygen supply, agitation or anything. But by only changing the recycle ratio, we can get different percentage of product conversion. These are the major advantages of the recycle bioreactors. Next, disadvantages of the recycle reactor. The first point, fixed cost is high because of higher volume of reactor required and operating cost is also high due to recycle. Normally, the recycle reactor are used in many larger chemical industry. For example, petrochemical industry and other chemical industry. In order to getting larger volume of product at a short time or in a single batch. So, the cost of running is somewhat high. Then, the volume required is higher than plug flow reactor due to recycle. I already told the recycle reactor are used in larger industry. So, the input volume should be higher than compared to other normal plug flow reactors. Next, conversion is also less than plug flow reactor. That means when compared to plug flow reactor, the product conversion rate is somewhat less in recycle reactor. These are some disadvantages or limitations of the recycle reactor. Once again, some important key points of recycle reactor. Recycle reactors are used when the reaction is autocatalytic. Next, in a recycle reactor, The product stream from a plug flow reactor is divided and a portion is returned to the entrance of the reactor. Then recycling provides a means for obtaining various degrees of back mixing with a plug flow reactor. Then the recycled reactor requires lesser time for the conversion of substrate into product than compared to other bioreactor. Also, we can get different percentage of conversion by only changing the recycle ratio in the recycled reactor. These are some key points about recycled reactor. Thank you.